Hello everyone, welcome to my channel. I'm your instructor for Pearson Edexcel IGCSE and GCSE Sciences. In today's video, we'll be delving into the details of Pearson Edexcel IGCSE Biology Paper 1 from June 2023. I'll be covering more papers and specific content for each science subject in my upcoming videos. But right now, I understand that many of you are eager to dive into the June 2023 paper. So without further ado, let's get started. Okay, so question number 1, A part states all living organisms share characteristics. State 2 characteristics that all living organisms share. So, as we all know, they respire, they reproduce, they require nutrition, they excrete waste, they grow, they respond to the surroundings, which we call sensitivity. So you can write any two of these. Next part states some organisms are pathogens. Which of these organisms can cause a bacterial disease in humans? So we know the correct option is D. B part, viruses are pathogens but not living organisms. Describe the effect of a named virus that infects plant. Right, so the named virus is tobacco mosaic virus, and the effect is that it causes the leaves of the plant to turn yellow. Moving forward with the last part, give 3 differences between the structure of viruses and bacteria. So viruses have a protein coat. They have a protein coat. They are much smaller in size as compared to bacteria. They lack nucleus, cell wall, cytoplasm, cell membrane. They lack vacuole. Okay, so you can mention any 3 of these. With that, we are done with question number 1. Let's move to question number 2. The diagram shows part of a food web from an ecosystem. Using the information in the food web, draw a food chain that contains 5 trophic levels and includes a bird. Alright, so there are a couple of food chains that we can give. The first one is dead plants. Excuse me for the arrow. It's small, let's make it small, and plants, fungi, not plants. Next up we have small arthropods. Next up we have birds. So you can see here that it has 5 trophic levels. as a arrows there are 4 and there are 5 plants. The next one that you can mention is dead plant, make sure to put the arrows correctly. Bacteria, and finally birds. And the last food chain that you can write about is plants, worms, okay. So these are the 3 food chains, so you can mention any one of these. Moving forward, describe how the bacteria and fungi obtain energy from the organic matter. So there it has 3 marks. So first mark is for mentioning the process, which is saprotrophic nutrition. So this is, you have gained 1 mark after mentioning this. Then you'll explain the process. They release digestive enzymes onto extracellular digestive enzymes. Make sure to mention extracellular. Onto dead material and convert the material into small soluble molecules, and then they digest it. Alright, so B part states a pesticide gets into this ecosystem. The pesticide kills small and large arthropods. Explain how this affects the population of birds and the population of worms that eat roots. Okay, so as you can see, small arthropods eat worms. So as the number of arthropods will decrease, the number of worms will increase because there will be fewer predators to eat them. And birds' population will decrease as less food will be available to them. So let's write this down. More worms as fewer predators will be there to eat them. Less birds as less food will be available for them to eat. So moving forward to C part, scientists use 3 different traps to collect some soil animals. These traps are a basket trap, a pitfall trap, and a cul-de-sac trap. The number of animals collected by each trap are counted each day. The animals collected are classified into 3 types: springtails, mice, and large arthropods. Graph shows the scientist results. Discuss the number of each animal type collected by the 3 traps. Refer to the scientist results in your answer. Alright, so this is worth 5 marks, so we need to attempt it very carefully. So let's mention the points here. More mites collected in each type of trap. Fewest arthropods collected in each type of trap. Next is more mites collected by cul-de-sac trap. Little difference in mite between cul-de-sac and basket trap. Fewest mites collected by pitfall trap. Most springtails collected by basket trap. Little difference in springtail numbers between cul-de-sac and basket trap. Fewest springtails collected by pitfall trap. And lastly, number of arthropods almost equal in all the 3 traps. Alright, so these are 8 points. You can mention any 5 of them. With that, we're done with question number 2. Let's move to question number 3. The diagram shows a plant cell with some structures labeled. Which structure is the cell vacuole? So that's T. Next part, which structure is the site of photosynthesis? So that's chloroplast, which is P. Moving forward, which structure is the cell wall? So that is S. Which structure U is the site of protein synthesis in the cell? What is the name of structure U? So protein synthesis is the function of ribosome. So C. B part, a cell is shaped like a cube. Each side has a length of 0.053 mm. Calculate the surface area to volume ratio of this cell. Give your answer in the form n/1. Alright, so the formula of volume is length cube, or length into length into length. So volume will be 0.053 into 0.053 into 0.053, which is if you do it on calculator, you will get an answer of 0.000148,877. So this is the volume. Talking about the surface area, the formula for surface area is 6 into length into length. So 6 into 0.053 into 0.053, which is 0.016 854. So this is the surface area of the cube. Now we have to find surface area to volume ratio. So surface area is 0.016854 and the volume was 0.000148877. So you will divide both of these values and the answer you will get is 113.21. So this is the final answer, 113.21. So with that we are done with this part. C part states animal cells, unlike plant cells, do not have a cell wall. Explain how this difference affects red blood cells when placed in distilled water. Okay, so red blood cell is an animal cell and placed in distilled water means that it is placed in high water potential. So water will move from high water potential to low water potential by the process of osmosis. So it will enter red blood cell. Since red blood cell lack cell wall, so it will not be able to prevent overexpansion, leading to red blood cell being burst. So this is the full answer, that water will enter by osmosis, and since red blood cell lacks cell wall, so it will burst. Next is explain how the difference affects red blood cell when placed in a concentrated salt solution. So concentrated salt solution means low water potential and high water potential inside red blood cells. So water will leave red blood cells by the process of osmosis. Red blood cells will push cell membrane away from cell wall and they will be shriveled. So this is the so question number 4 states the diagram shows a human reflex response to touching a hot object. Some of the structures in the reflex arc are labeled. Give the name of this reflex response. So this is withdrawal response, or you can even mention involuntary response. Next part, draw an arrow on the diagram to show the direction of the nerve impulse at point X. So point X, so that is motor neuron is taking it, so it will be in this direction. This is arrow. Okay, so this it will load towards the muscle. Next part, which structure detects the stimulus? So stimulus is detected by obviously E, because that is a finger. Which structure is in motor neuron? So it is I. Next part, a neuron is 1.10 meter in length. The speed of the nerve impulse in this neuron is 120 meters per second. Calculate the time in seconds for the impulse to pass along the neuron. Give your answer in standard form. Okay, so the formula for speed is distance divided by time. So since we have the speed, we want to calculate the time, so we'll divide distance by speed to get the time. So distance is 1.10 and the speed is 120. So if you divide this, you will get the answer 0.0092, and then you have to write it in the standard form. So 9.2 into 10 raised to the power minus 3. So this is our final answer. 9.2 into 10 raised to the power minus 3. So next part, describe how the impulse passes from neuron to neuron in the reflex arc. So neuron passes to synapse, neurotransmitters, diffuse along the synapse and passes the neurons. So this is the answer. Next part, pain has a survival function. If a person damages their ankle, they sense pain. Explain how sensing pain benefits an organism. So obviously, if you are able to sense pain, you can take your hand back and prevent movement. So it prevents movement and obviously from further damage, and prevent further damage or injury. Next part and the last part, some medicines are used to reduce pain. These medicines work by preventing communication between the injured ankle and the brain. Explain which components of the nervous system may be affected by these medicines. Okay, so they prevent communication between the ankle and the brain. So ankle, we have the receptors over there, so receptor cells will not work, so it will prevent detection of pain by receptor. Then it will prevent transmission of impulse in sensory neuron, then in relay neuron, because if it will not go in sensory neuron, then it will obviously not go in relay neuron. And it will also prevent detection of synapse transmission. Moving forward to question number 5. Red blood cells and white blood cells are two of the components found in human blood. State two components of blood. So plasma and platelets. B part, some white blood cells destroy pathogens. The diagram shows 4 stages in this process. Describe what is happening in each stage of this process. So in stage 1 and 2, phagocyte is engulfing the bacterium. In stage 3, it is releasing digestive enzymes to digest it, and in stage 4, the products of digestion are removed. So with that, we are done with this. Part C, the picture shows the human red blood cell. The actual diameter of this human red blood cell is 8.1. Determine the magnification of the picture. So magnification has the formula image size divided by actual size, which is measure the image size, that is around 5.7 or 5.5 to 5.9, the range. Any answer in between is acceptable. So we'll convert 5.7 centimeter by multiplying it with 10,000 so that the units are same. So it will be 57,000. Now we'll multiply. Magnification is equal to 57,000 divided by 8.1, which is the image actual size. And the answer that we are getting is it's 7,307. So that's the final answer. So this X is the sign of magnification. This part says patients with reduced number of red blood cells are described as anemic. In an investigation, scientists measure the percentage oxygen saturation of the blood and the rate of tissue deoxygenation. The rate of tissue deoxygenation is a measure of how fast oxygen is lost from tissues. They use 4 groups of patients: non-anemic patients with normal blood flow, anemic patients with normal blood flow, non-anemic patients with slow blood flow, and anemic patients with slow blood flow. The table shows the scientists' results. Discuss the relationship between anemia, blood flow, percentage, oxygen saturation of blood, and rate of tissue deoxygenation. You should refer to data in the table and use your biological knowledge in your answer. Okay, so let's make notes. Normal blood flow, percentage of oxygen is higher than slow blood flow in non-anemic patients. Normal blood flow, rate of deoxygenation is higher in non-anemic patients. Percentage of oxygen is lower in anemic patients, in normal and slow blood flow. Next up is greater difference in percentage oxygen between non-anemic and anemic patients in slow blood flow than normal blood flow. Next up we have fewer red blood cells to carry oxygen in anemic patients. Anemic patients tissues deoxygenate faster. Next up we have little difference in deoxygenation between normal and slow blood flow patients. And the last point which we can mention is other factors like smoking, activity levels, and diet can also affect the results. So these are a couple of points. You can mention any 5 of these to gain full marks. With that, we are done with question number 5. Let's move to question number 6. Many characteristics of pea plants are genetically controlled. One of these characteristics is flower position. Flower position can either be axial or terminal. In a first cross, scientists crossed pea plants with axial flowers with pea plants with terminal flowers. The first cross produced 1120 offspring plants. All of these offspring plants had axial flowers. Use a genetic diagram to show the genotypes of the parent plants, the gametes they produce, and the genotypes of the offspring plants. So, the genotypes of the parents are AA and AA, small a and capital AA. Gametes that they produce are A and A, and the genotype of offspring is all are A and A. So this is the answer to this. You can make a genetic diagram to check, but this is mainly the points. B. The scientists allowed the offspring from the first cross to self-fertilize. This second cross produced 858 second generation plants. 608 of the plants had axial flowers and the other plants had terminal flowers. Calculate the ratio of plants with axial flowers to plants with terminal flowers. Give your answer in the form n/1. Okay, so 858 second generation plants we have. So 858 minus 608, which is 250, and then we'll divide, basically we'll write it in ratio 608 ratio 250, which is 2.43. so this is our final answer, 2.43 ratio 1. Next part states, explain why the ratio of plants with axial flowers to plants with terminal flowers is different from the expected ratio. So expected ratio is 3:1. So there may be random fertilization. Random fertilization could have caused this. So now the first mark is for expected ratio, the second is random, and the third is fertilization. Simple fertilization will give you 1 mark. Last part. Scientists want to discover if plants with axial flowers produce more seeds than plants with terminal flowers. Describe an investigation to discover whether plants with axial flowers produce more seeds than plants with terminal flowers. Include experimental details in your answer and write in full sentences. Okay, so whenever we are planning any investigation, the best way is to first of all mention the independent variable, basically to use comps. If you know this is the technique. C, for you are going to mention what are you going to change. So we will change the plants with axial flowers and with terminal flowers. Then O is for organism, so we'll use same species of plant, same size and same age of plant. Next is R, which is repeat the investigation and calculate the mean for reliable results. Next up is M, so we'll measure the number of seeds produced after a stated time period. So you can give after 2 days. And the last one, S, means use same temperature, same mineral ions and same conditions. So this is the investigation. With that we are done with the investigation. And question number 6. The next question number 7 states, lipids digest lipid into fatty acids and glycerol. The fatty acids change the pH of the solution. A student uses this method to investigate the effect of temperature on the activity of lipids. Step 1. Label a test tube with the temperature 20 degrees Celsius. Step 2. Add 5 drops of the indicator to the test tube. Then add 5 centimeter cube of milk to the test tube, and all of these steps. So, question 7A part states, state why the student leaves the test tube and the beaker in the water bath for 5 minutes in steps 6 and 7. Okay, steps 6 and 7. Place the test tube in a water bath and place the beaker container in the same water bath for 5 minutes. So he places to maintain temperature. That's the function of water baths. So B part, give the dependent variable in this investigation. So as we can see that the investigation is basically to see the effect of temperature on the activity of lipids. So we will write the dependent variable time taken. Dependent variable is a variable that changes itself. Time taken to lose pink color, like we can't change it, right? So that is a dependent variable. Next is state one variable the student controls in their investigation. So that is volume of lipids, or you can say volume of milk, or you can say time taken in water bath, time left in water bath. Next up we have stage to suggest the purpose of the phenolphthalein indicated in the investigation. So the purpose is to show that all of the lipid has digested. Okay, so the part states, the table shows the student's results. Plot a line graph to show the effect of temperature on the time taken for the contents of the test tube to lose the pink color. Use a ruler to join your points with straight lines. So you have to make the graph. Okay, so you'll make the graph. You'll take temperature on x-axis and time taken on y-axis, because the independent variable that you can change is always on the x-axis. So that will be temperature, and on y-axis is time taken, and then you'll make the line graph. Some rules about graphs are that make sure that it covers your 50% of the graph your scale covers. So choose such scale, and rest is that try to make it from pencil so that it is neat and clean and you have the margin of making mistakes and correcting them. So you can make this. Next up is explain why increasing temperature affects the time taken for the contents of the test tube to lose the pink color. Okay, so as you can see, as we increase the temperature, the time taken decreases until 40. After that it increases. So what happens is that as temperature increases, the kinetic energy increases. Enzymes, there are more collisions, and enzymes work faster until optimum temperature, which is the temperature, I guess 40. Yes, until 40 degrees. After that, enzymes denature. The active site changes and substrate no longer binds, leading to decreased activity and increased time to lose the pink color. So with that, we're done with question number 7. Moving forward to question number 8. Yeast can be used in experiments to investigate the effect of different concentrations of glucose solution on the rate of anaerobic respiration. The diagram shows apparatus used to measure the rate of anaerobic respiration in yeast. Give the function of the water bath. So the function of the water bath is to maintain the temperature or to keep the temperature constant. It's the same thing. Okay, so next up we have liquid A can be used to identify the gas released during anaerobic respiration by the yeast. Explain which substance can be used as liquid A. So the gas released is carbon dioxide in anaerobic respiration. So carbon dioxide can be identified using lime water. It turns milky. It will be used milky. Fine, it turns milky. So next up we have a student wants to accurately measure the rate of anaerobic respiration in yeast using this apparatus. State what additional apparatus they would require. Okay, so they would require a stopwatch to measure the time and a gas syringe to measure the volume of the gas. So you can mention any one of these because it's 1 mark question. D part, the process of anaerobic respiration is sometimes referred to as fermentation. Industrial fermenters are used to grow microorganisms. These microorganisms produce penicillin, an antibiotic that is used to treat bacterial infections. Explain how named conditions in an industrial fermenter are controlled. Okay, so an industrial fermenter, temperature is controlled using temperature monitors to provide an optimum temperature for enzymes to work. Next up, we have pH is maintained using pH probes to provide an optimum pH for enzymes to work. Moreover, stirrer paddles are used to mix the contents. Oxygen is provided using air inlet for aerobic respiration. Furthermore, what else do we have? Temperature, pH, oxygen. Air filter is used to prevent the contamination from bacteria. So that's all. Moving forward to the last part, some antibiotics are becoming less effective at controlling bacterial infections. Explain how bacteria have evolved so that antibiotics are less effective. So bacteria have basically developed resistance. So what happens is, due to mutation, there is variation and bacteria has developed resistance to antibiotics. Bacteria have developed resistance to antibiotics. And resistant bacteria is more likely to survive, then non-resistant bacteria. It reproduces and pass on the passes on the alleles. So with that, we're done with question number 8 and question number 9 and the last question for this paper. Genetically modified bacteria are used to produce the hormone insulin. Explain how these genetically modified bacteria are produced. So what happens is human insulin from, you can mention from human chromosome if you want, or you can even skip, is cut using the restriction enzyme then using the same restriction enzyme leaving it with sticky ends. Using the same restriction enzyme, plasmid from bacteria is cut and isolated. DNA ligase enzyme joins both of these into a single strand of DNA. The recombinant plasmid is then inserted back into the bacterium, and the bacteria has now been genetically modified. So this is the process. Okay, so that is it. Next up, explain the role of insulin in the human body. So insulin causes the liver to take up glucose and convert it into glycogen, so lowers blood glucose levels. So with that, we are done with this part. C part. Some people are unable to produce insulin. This condition is called diabetes mellitus. People with diabetes mellitus control the condition by using insulin injections, controlling their diet, and monitoring how much they exercise. Explain why the insulin is injected rather than taken in the mouth. So insulin is a protein which needs to be digested by the pepsin enzyme in the stomach. So basically it's large enough so it cannot be taken into the mouth. That's why it needs to be digested. Then state why people with diabetes need to control how much they exercise. So people with diabetes, they have more respiration. When they exercise, they respire more, so they use up more glucose, which can result into lower blood glucose levels, basically too low. Can if they exercise too much, blood glucose levels can drop significantly, leading to a lack of glucose. Okay, so next up we have state how people with diabetes may need to modify their diet compared with people who do not have diabetes. So people with diabetes are not able to absorb glucose, so they need to control blood glucose levels. Need to control blood glucose levels. So with that we are done with question number 9 and paper 1 of June 2023. I hope you all enjoyed the paper and it genuinely helped you all. If you have any questions, please feel free to leave in the comment section below, and I'll see you soon with another video. Take care.